Hello everyone, welcome back to Mahi's Life Science channel. In this video, we will discuss about the factors affecting enzyme activity. First, we will see what are the factors which affect the enzyme activity. They are temperature, pH, substrate concentration, and the presence of inhibitors or activators. Biochemical reactions are necessary for the growth, repairing damaged tissue, and obtaining energy that take place in all living organisms, and these reactions are called as a metabolism, and this metabolism will happen all the time in living organism. If the metabolism stop working, this lead to the death of the organism. All the reactions that occur in living organism require high activation energy. To reduce the cell's consumption of energy, there is a catalyst to ensure that the chemical reaction occur rapidly and reduce the activation of energy. This catalyst is the enzyme. Enzyme are the biological catalyst which is made up of a large protein molecules. They speed up the chemical reaction inside the cell. The enzyme is made up of the combination of amino acid in which for a chain of polypeptide occur between each other. Enzymes are similar to other chemical catalysts. They participate in the reaction without getting affected. In other words, we can tell enzyme, they speed up the chemical reaction inside the cell without getting consumed. Enzymes are mainly affected by the pH, that is hydrogen ion concentration, and the temperature. Enzymes are highly specific compared to other catalysts, and each enzyme is specialized for one reactant substance. The reactant substance is called as the substrate, and it is specialized for one type of reaction or a few reactions. Enzyme lower the activation energy required to get the reaction started. Collectively, they are the most important property of the enzyme. There are several factors affect the speed of an enzyme action, such as concentration of the enzyme, concentration of the substrate, temperature, hydrogen ion concentration, and the presence of the inhibitors. First one is concentration of enzyme. As the concentration of the enzyme is increased, the velocity of the reaction proportionately increases, and this property is used for determining the activities of serum enzyme during the diagnosis of diseases. Second one is concentration of substrate. In the presence of a given amount of enzyme, the rate of enzymatic reaction increases when substrate concentration is also increases until a limiting rate is reached. After further increase in the substrate concentration produces no significant change in the reaction rate. At a particular point, so much substrate is present that essentially all of the enzyme active site have substrate bound to them. In other words, we can tell the enzyme molecules are saturated with the substrate. That is, whatever the active site present in the enzyme which is completely occupied. The excess substrate molecule cannot react with the substrate already bound to the enzyme as reacted and been released. Third one is effect of temperature. The protein nature of the enzyme make them extremely sensitive to normal changes. The enzyme activity occur with a narrow range of temperature compared to ordinary chemical reaction. As we have seen, each enzyme have a certain temperature at which it is more active. This point is called as an optimal temperature, which ranges between 37 to 40 degrees Celsius. The enzyme activity gradually lower as the temperature rises more than the optimal temperature until it reaches a certain temperature at which the enzyme activity stops completely due to the change in its natural composition. On the other hand, if the temperature lower below the optimal temperature, the enzyme activity also lowers until the enzyme reaches a minimum temperature at which the enzyme activity is very least. The enzyme activity stops completely at 0 degrees Celsius, but if the temperature rises again, then the enzyme also gets reactivated once again. Effect of pH. The potential hydrogen is the best measurement for determining the concentration of hydrogen ion in a solution. It also determines whether the liquid is acidic, basic or neutral. Generally, all liquid with a pH below 7 are acids, whereas liquid with a pH above 7 are bases or alkalines. Liquid with pH 7 are neutral and they equal the acidity of pure water at 25 degrees Celsius. We can able to determine the pH of any solution using the pH indicators. Enzymes are protein substances that contain acidic carboxylic group and basic amino groups so that the enzymes are affected by changing the pH value. Each enzyme has a pH value that is worked at with maximum efficiency called the optimum pH. If the pH is lower or higher than the optimal pH, the enzyme activity decreases until it stops working. For example, pepsin work at low pH, that is, it is highly acidic, while trypsin work at high pH, that is, it is basic. Most enzymes work at neutral pH 7. Effect of activators. Some of the enzymes require certain inorganic metallic cations like magnesium, manganese, zinc, calcium, cobalt, copper, sodium, potassium for their optimum activity. Rarely, anion are also needed for enzyme activity. Example, chloride ion for amylase activity. Thank you. Subscribe and follow for more videos in microbial physiology and metabolism.